Good afternoon, everybody, respected faculties and all my dear colleagues. Myself, Dr. Aman Shakir, Junior Resident 2, Department of IHBT, MGM Medical College, Indore, will be presenting a seminar on Hardy-Weinberg principle and its application in blood banking. And moderator is Dr. Ramu Thakur, sir, Associate Professor, Department of IHBT, MGM Medical College, Indore. Overview. This is the overview I will be covering today. Introduction, population genetics, Mendel law of inheritance, Hardy-Weinberg principle in detail. Introduction. The characteristics making each blood group system unique are antigen structure and locations on the red cells, and the antibodies they elicit, and last, the genetic control of antigen expression on red cell membrane. These antigens differ depending on the inheritance of the blood group genes. So, which gene is inherited, it will produce its protein, and that protein will generate its blood group system. A blood group system, what is blood group system? It is composed of antigens which are produced by alleles at a single genetic locus or at loci so closely linked that genetic crossing over rarely occurs. What is gene? It is a unit of inheritance encoding protein and its basic unit for inheritance of a trait. It is a segment of DNA that encodes a protein, and genetic information is carried on double strand of DNA known as chromosomes. These chromosomes contains alleles. These are alternate forms of gene at a given loci on chromosome. For example, ABO are alleles on ABO gene locus. And what is the meaning of antithetical? Opposite antigens encode at the same locus. For example, in KL system, KPA and KPB. What is polymorphic? It is refers to the two or more alleles at a given locus. When it results from change from one nucleotide, it is referred to as single nucleotide polymorphism, SNP. For example, Rh blood group system, it is highly polymorphic because of many alleles. Now coming to phenotype and genotype. What is phenotype? Phenotype is the physical expression of inherited trait, that is antigens present on all red cell expressed. What is genotype? Actual genes which are inherited from each parent that controls what antigens may be expressed on the cell. For example, in phenotype A, genotype can be capital A or capital A O. Since O is no detectable product, only A is expressed. The frequency of phenotype in a population depends upon degree of polymorphism within a blood group system. For example, Rh blood group is highly polymorphic. It has around 61 alleles, antigens of it. Now coming to population genetics. The study of distribution patterns of genes and of the factors that maintain or change gene or allele frequencies. A basic understanding of population genetics, probability and application of algebraic calculation is important in transfusion medicine, where knowledge can be applied to predict the likelihood of compatible blood for a patient who has made antibody or antibodies to red cell antigens. It describes how a combined phenotype calculation is needed to locate compatible donor units for patients with multiple red cell alloantibodies. What is prevalence? Prevalence is used to describe the occurrence of a permanent inherited characteristic at a phenotypic level. For example, a blood group antigen. What is frequency? Frequency is used to describe prevalence at the genetic level, that is occurrence of allele in a population. Incidence is used to describe the rate of occurrence in a population of a condition that changes over time, such as disease. Population genetics is concerned to blood banking, which includes Mendel laws of inheritance, Hardy-Weinberg principle, and inheritance pattern. Since we are dealing with Hardy-Weinberg principle, it is important to know about Mendel laws of inheritance, because it is carried forward by Hardy-Weinberg to understand the genes pattern. The Mendel laws of inheritance. The understanding of inheritance was made possible by Gregor Mendel, Austrian monk and mathematician, who formulated certain laws to understand the inheritance. What is inheritance? It is defined as a process of how a child receives genetic information from parent. The whole process of heredity is dependent upon inheritance, and it is the reason that offsprings are similar to the parents and the members of the same family possess similar characteristics. Mendel conducted hybridization experiment on garden peas to study physical traits and how they are inherited, to call them elemental within the cell, which is actually a gene. After that, actually it is a gene which is present in chromosome and its nucleus of the cell. He chooses distinct characteristics of peas like flower color, seed, and he conducted a cross pollination and to pea lines to see the lines lineage in pea families. He conducted two main experiments: monohybrid cross and dihybrid cross. Monohybrid cross, he took two pea plants, one is tall and one is short, and crossed them. He found that first generation offspring were tall and call it as a F1 progeny. Then he again crossed F1 progeny to obtain a tall and short plants in a ratio of 3:1. And he conducted this experiment and other contrasting traits also, like green pea versus yellow pea, round, wrinkled. In all cases, he found the result were similar, and he formulated the law of segregation and dominance. In that experiment, he took the first generation was pure parental and call it as a P1 generation. Either it were homozygous, capital R dominant trait, or white flowers, small r recessive trait. In first generation, like we call it first filial generation, all flowers were red. But again, after crossbreed in second filial F2, he found the three flowers were red and one flower was white, because only white color, only homozygous small r, it will produce white. Otherwise, all capital R, either capital R or capital R small r, it will produce red only. And he formulated the law of dominance, like hybrid offsprings will only inherit the dominant trait in the phenotype. These alleles are suppressed, which are called as recessive traits, while the allele that are determine the trait are known as dominant traits. In dihybrid cross experiment, Mendel considered two traits having two alleles. He crossed wrinkled green seeds and round yellow seeds and observed that all the first generation were round yellow. This means that dominant traits were round shape and yellow color, not the wrinkled and green. He then self-pollinated the F1 progeny and obtained four different traits: round yellow, round green, wrinkled yellow, and wrinkled green seeds in ratio 9:3:3:1. From this, he proposed law of independent assortment. It states that during production of gametes, two copies of each hereditary factor segregate so that offspring acquire one factor from each parents. In other words, allele, all alternate form is gene, pairs segregate during the formation of gamete and reunite randomly during the fertilization. This is the experiment, here it is a first generation, and he found in F1 progeny only round and wrinkled seeds. And in second generation, F2 generation, he found 9:3:3:1 ratio. 9 is round and yellow, 3 is round and green, 3 is wrinkled and yellow, and the last one is the recessive one, which is wrinkled and green. Again, this is the genotypic expression of all the alleles that are arranged in F2 generation, while phenotypic presentation is 9:3:3:1. What is law of segregation? Second law. This segregation law states that during the production of gametes, two copies of each hereditary factor segregate so that offspring acquire one factor from each parent. So this is segregation. Pairs segregate during the formation of gamete and reunite randomly during the fertilization. Law of independent assortment. It is a third law. It states that pair of traits segregate independently of another pair during gamete formation, but these individual hereditary factors assort independently. They characterize their features independently. Different traits get equal opportunity to occur together. This is the classification, like in MNS system. Here we can see M and N. They, although they are opposite to each other, antithetical, but still they are presenting in progeny, successive progeny. So neither of the one is suppressed, both M and N have equal opportunity to express themselves in successive progeny. This is law of independent assortment. Now, Hardy-Weinberg principle. G.H. Hardy, a British mathematician, and W. Weinberg, German physician. What they studied in population genetics, they form a principle also known as equilibrium. They state that allele and genotype frequencies in a population will remain constant from generation to generation in the absence of other evolutionary influences. The allele or gene frequency is the proportion of one allele relative to all alleles at a particular gene locus in a given population at a given time. So the sum of these allele frequencies must be equal to 100% or 1 in algebraic equation. So he given that if two alleles are there, the sum of these two alleles should be equal to 1. The summation that. So that was about gene frequency or allele frequency. Allele frequency, gene frequency is P plus Q is equal to 1. But in genotypic frequency, we consider genotype. So number of individuals with a given genotype or total divided by total number of individuals sampled in a population. It is the genotypic frequency. The gene frequencies tend to remain constant from generation to generation in any relatively large population unless they are influenced by factors such as selection, mutation, migration, or non-random mating. The gene frequencies reach equilibrium and can be expressed in algebraic terms. P square plus 2pq plus q square is equal to 1. If, for example, if two alleles are capital A and small a, have gene frequencies of P and Q respectively, the homozygous and heterozygous are present in population in the following proportion. Capital A is P square, capital A small a is 2pq, and small a is q square. That is recessive one. If gene frequency of one allele is known, Q can be calculated by P plus Q is equal to 1. If P is known, we can calculate by Q is equal to 1 minus P. We can calculate P if one allele frequency is known. So P, this is the Punnett square chart which shows that P square plus 2pq plus q square is equal to 1. This one. This again, what I have said, P is a dominant allele, Q is recessive allele, and some of it is plus 1, and is the genotypic one. Frequency of homozygous dominant genotype is P square, frequency of heterozygous genotype is 2pq, and frequency of homozygous recessive genotype is q square, that is equal to 1. So when we consider allele, this formula is used, P plus Q is equal to 1, and when we are considering the genotype, we are considering the P square and Q square formula. What are the criteria for the use of Hardy-Weinberg formula? The population studied must be large, mating among all individuals must be random, mutations must not occur in parent or offspring, and there must be no migration, differential fertility or mortality of the genotype studied. But there is drawbacks. This condition is not fulfilled because collecting sample data from large population is not feasible, mating is not always random, gene mutations occur, some mutations affect the phenotype of an individual, and there is mixing of population on a global scale leads to gene flow on a constant basis. What is the use of Hardy-Weinberg in blood banking? If for a given population, if the prevalence of one genetic trait for such as red cell antigen is known, Hardy-Weinberg equation can be applied to calculate both allele and genotypic frequencies. The formula which I have shown before. For example, to find the frequency, example 1, to find the frequency of Rh antigen, capital D in a given population, we can assess the genotype also, like absence of complete RHD gene denoted as small d. So it means that both capital D and capital D small d will appear as D positive. To calculate the P, we count the number of individuals who have corresponding phenotype, that is D positive, which is capital D and capital D small d, and divide it by total number of alleles. Then count for Q. So we found the formula P plus Q is equal to 1. If P is known, we can calculate Q also. Ratio of homozygotes and heterozygotes is determined by p square plus 2pq plus q square is equal to 1. Now, if we assess 1000 random blood donors were tested for capital D, then capital D positive phenotype will be 84%. We denoted as capital D and capital D small d genotype, which will be cap D positive only because capital D is dominant allele. So P square is equal to capital D, 2pq, capital D small d, it will equal to 0.84. If we found that 84% is there, then negative 1 will be 16%. Q square is equal to 0.6, and after calculation it is coming at 0.4. Now we come to know about Q, we can find P also. 1 minus 0.4 is equal to 0.6. For 3 allele system, the equation can be expanded, but it will form like this one. Example 2, we can find in Indian population, we can find the frequencies of 2 alleles encoding the antithetical antigens of KL can be determined. If we consider allele, KL12 is P and Q. If we consider genotype, KL12 is P square and Q square. And both the genotype, if I express genotype, then 2pq. Now, if we know that in Indian population, the K antigen is expressed on 4.4% of people of Indian population. So we can, by this capital K, we can consider that KL1 or K positive will be P square plus 2pq, and that will be 0.44. It is percentage converted into unit. So that's why KL0202, that means recessive one will be KL negative, it will be recessive one will know that Q square it is there. Then Q square will be 1 minus the dominant one. So 1 minus 0.044 is equal to 0.956. So by this formula we can conclude that Q is equal to 0.977. From this we can calculate P value. P 1 minus 0.997 is 0.023. So this is P value. We can conclude by prevalence of genotype, we can conclude that P is 0.023 and Q is 0.977. Once the allele frequencies are calculated for both KL1 and KL2, it is possible to calculate the percentage of both KL positive and KL capital K positive and small k positive alleles. Capital K includes both capital K plus or small k plus and capital K plus small k minus. And small k positive includes both capital K plus and small k plus and capital K minus small k negative. Here I have highlighted what they are mentioning here, capital K plus and small k plus. Since capital K plus and small k minus is actual the P, so it will be considered as 0.23, which is P value. And since capital K is negative and small k is positive, it is actually the Q1, which I have found, which we will found at 0.977 Q value. And both of them are positive here, capital K positive and small k positive. It is 2pq, actually the 2pq. So we can put 2pq here, calculate it as 0.044. So we found that P value is here this and Q is this and 2pq is this. Now we can put in the formula of P square plus 2pq and Q square, and we can calculate the genotypic frequencies also. So prevalence of K positive, only K positive will be 2pq and P square, since P square is whole pure homozygous one and PQ is heterozygous one. So we can calculate the prevalence of K positive as 4.4%. So from genotype frequency prevalence, we can find allele distribution also, and from allele distribution we can find genotypic prevalence also. We can find vice versa. Like from 4% 4 we came to conclusion of P value, and from P value we can came to conclusion of 4.4, like this. Similarly, in prevalence of K minus, it is Q square, the homozygous one, and 2pq, the heterozygous one, which contains small k. So we can, by concluding, we can conclude that K negative prevalence of K negative, small k negative is 99.4%. Yes, ma'am. mistake. Small k positive it is. So prevalence is small k positive will be 99.4 and prevalence of capital K positive will be 4.4. Now three possible genotype frequencies will be as follows. From this homozygous one, KL1 and KL1 homozygous, it is actually the P square. P we will know, so we can by squaring it we come to percentage of possible frequency of this will be this, and frequency of KL1 and 2, which is it is a homozygous one. So 2PQ by putting the value of P and Q, we conclude that it is 0.044, and frequency of KL22 like homozygous recessive, so it is Q square 0.977 square. It is come to known as 9.956. So by adding to this 0.956, 0.044, 0.000529, we come to total will be 1 only. So we can find allele frequencies and genotype frequencies from Hardy-Weinberg formula. What is phenotypic prevalence? It is determined by testing red cell from a large random sample of a people of same race or ethnicity with a specific antibody, then calculating the percentage of positive and negative reactions. What we are doing here, like positive and negative reaction only. For example, for antithetical antigens, the sum of percentage for the prevalence should be equal to 100%. For example, if we assess 1000 donors and we found that 800s are positive with NTC and 200s are negative with NTC. So prevalence of C plus small c plus will be 80% and small c minus will be 20%. So for this minus 20%, we can conclude that 1 in 5 units should be compatible with the serum plasma of patient who has made NTC. Now, how we can calculate the antigen negative phenotype? To provide the patient provide the blood to patient with antibodies which are directed against the red cell antigens, equation can be used to estimate the number of units that needs to be tested. The prevalence of each of the individual antigens are multiplied together if the antigens are inherited independently of each other. It is not valid when antigens are coded by alleles that are closely linked and inherited as haplotypes. For example, MNS system, or reside on the same carrier protein, capital C small c and capital E small e. So this is the formula which has been taken from DGHS. For finding the number of antigen negative units required, we can divide it by 1 minus prevalence, phenotypic prevalence of antigen A of first L of antibody into multiplied by 1 minus phenotypic prevalence of antigen of second antibody in n times. For example, the prevalence of donors of Indian population who are capital K positive is 4.4%. So capital K negative will be 95.6, this one, and capital S positive is 54.2%. So capital S negative will be 100 minus 54.2 is 45.2, and JKA positive is 80.8%. So JKA negative will be 19.2%. If we want to give negative the percentage of such negative donors for each antigen is expressed as decimal and is multiplied. So 0.95, 0.45 and 0.919. If we want to find the unit which is negative for all these three antigens, we need to multiply the negative prevalence of these antigens. So we found that 0.0812 is the is the unit, and it is expressed in percentage. So we have multiplied by 100. It is come as 8.12%. So 8.12 expressed as occurrence will be 8.12 1 in 12 units. So approximately 1 in 12 ABO compatible RBC units are expected to be capital K minus, capital S negative and JKA negative. So if this patient requires 3 units, then we can multiply it by that factor, and it is coming as 32 units, 36 units. So to find 3 units compatible, we need to test 36 units so that we can get probability of getting this is possible. Thank you. Exercise. Anybody can.